glycogenesis, meaning glycogen synthesis. So glycogen synthesis occurs from glucose in the cytoplasm, and this synthesis requires ATP, UTP and glucose. So the entire process of glycogen synthesis can be described under 4 steps. First is the activation of UDP glucose. Second is the initiation of glycogen synthesis. Third is the formation of linear units in the glycogen, and fourth is the formation of branches in the glycogen. First step is the activation of glucose or the synthesis of UDP glucose. So in the presence of the enzyme glucokinase in liver, whereas hexokinase in muscle, this glucose gets phosphorylated to form glucose 6-phosphate. Now this glucose 6-phosphate is converted to glucose 1-phosphate in the presence of the enzyme phosphoglucomutase. Next, this glucose 1-phosphate combines with UTP in the presence of the enzyme UDP glucose pyrophosphorylase to form UDP glucose and high energy phosphate molecule. So the formation of UDP glucose is important in this step. This is how there is synthesis of UDP glucose and activation of the glucose molecule. So next is the initiation of glycogen synthesis. For the initiation of glycogen synthesis, a primer is required. Now what is a primer is? A primer is the first point where the actual synthesis starts. So here in glycogen synthesis, glycogenin or a small part of glycogen itself will act as a primer, and to this point the further attachment of glucose unit take place. Now this glycogenin is a dimeric protein which is made of 2 subunits, and it has an amino acid tyrosine, and the hydroxyl group of the amino acid is the site where the glucose unit is first attached. So the UDP glucose that is synthesized, this UDP glucose, once it is synthesized, the glucose unit from this UDP glucose is transferred to the hydroxyl group of the tyrosine of glycogenin in the presence of the enzyme glycogen synthase. So glycogen synthase helps in the transfer of glucose units from UDP glucose to the glycogenin or glycogen primer, and later glycogenin acts as an acceptor for the rest of the glucose units. So first, initially when the glucose unit is attached to the primer in the presence of the enzyme glycogen synthase, later the entire molecule acts as an acceptor for the rest of the glucose unit. So the process of addition of glucose unit is called as glycosylation, and this continues till 7 units of glucose are added. So this process of addition of these glucose molecules in a linear fashion, this will continue till 7 glucose units are added. Next is formation of linear units. So the glycogen synthase transfers glucose from UDP glucose to the non-reducing end of glycogenin or glycogen primer to form alpha 1,4 linkages. So this is the glycogenin molecule, and the hydroxyl group of glycogenin or the tyrosine acts as the first acceptor of the glucose molecule, and this has one reducing end and one non-reducing end. And to the non-reducing end specially, the glucose molecules keeps on getting attached, and the linkages that are formed in the linear molecule are alpha 1,4 linkages. So glycogen synthase is the enzyme. It can only form the straight alpha 1,4 linkages, and the UDP that is bringing the glucose, once glucose is attached to the glycogenin, the UDP is released back and it is converted to UTP by nucleoside diphosphate kinase. For the formation of branches in glycogen structure, branching enzyme called as amylo 1,4 to 1,6 transglucosidase is required. This enzyme specially forms alpha 1,6 glycosidic bonds that serves as a branch in the glycogen structure. After the addition of few glucose units in a linear manner, this branching enzyme will transfer about 7 to 8 units of glucose from the non-reducing end and that is the end away from the primer, and it binds to any other side. So end away from the primer, this is the primer, and the end that is away from the primer is called as non-reducing end. So from the non-reducing end, about 7 to 8 glucose unit molecules are taken, and they are attached by alpha 1,6 glycosidic linkage, and the enzyme which helps in this attachment is amylo 1,4 to 1,6 transglucosidase. Now the new 7 to 8 glucose units are held in the linear structure by alpha 1,6 linkage. So the linear structure, this is the linear structure, and to this linear structure the new glucose units are held together by alpha 1,6 bond, and each glucose unit in the linear structure are held together by alpha 1,4 bond. After this process of branching, further new glucose units are added linearly to the newly formed branch by the enzyme glycogen synthase. So the glycogen synthase keeps on adding glucose units to this newly formed branched structure in a linear manner. These steps result in the formation of a highly branched tree-like structure of glycogen. So to summarize, first is the activation of glucose, where glucose in the presence of the enzyme hexokinase or glucokinase is converted to glucose 6-phosphate. Now this glucose 6-phosphate is converted to glucose 1-phosphate in the presence of the enzyme phosphoglucomutase. Next, the glucose 1-phosphate combines with UDP in the presence of the enzyme UDP glucose pyrophosphorylase to form UDP glucose. And this UDP glucose brings the glucose molecule to glycogenin, which acts as a primer. Primer is the initiation, is the starting point of the glycogen synthesis. So this glycogenin has an amino acid tyrosine, and the hydroxyl group of the amino acid tyrosine acts as the first acceptor of the glucose molecule. So UDP glucose transfers this glucose molecule to the glycogenin primer in the presence of the enzyme glycogen synthase. Glycogen synthase can add glucose molecule only in linear fashion, and this process of addition is called as glycosylation, and the branches, the linkages that are present in this linear molecule are alpha 1,4 bonds. And once a linear chain of 7 to 8 units is formed, a 2 to 4 unit is served as a branch, and the branching enzyme is 1,4 to 1,6 transferase, which will transfer 2 to 4 units of the glucose molecules to the branch point, and the branch point or the branched bond is alpha 1,6 bond. So further elongation at the non-reducing end by glycogen synthase making alpha 1,4 bonds continues, and further branching also continues to form a highly branched structure called as glycogen.